आज का बुक रीडिंग में आप सभी को स्वागत है। आज हम पढ़ने वाले हैं कॉम्प्रिहेंसिव टेक्स्ट बुक ऑफ मेडिकल फिजियोलॉजी वॉल्यूम 1 से बाय जी.के. पॉल। ठीक है, तो प्रीवियसली हमने स्मूथ एंडोप्लाज्मिक रेटिकुलम तक आई थिंक हमने कवर कर लिया था। ठीक है, नाउ आज हम पढ़ेंगे गोल्गी apparatus। गोल्गी apparatus सिंपली रिटन एज GA। Golgi apparatus is also known as Golgi complex or ये क्या है? डाइसाइटोसोम या क्या? ओके, 1 मिनट। DICTYOSOME डिक्टोइजोम। ओके, थोड़ा वेट कीजिए। डाइसाइटोसोम या फिर यार जिस वक्त इन लोग जब भी कोई प्रॉब्लम टाइम में मारो AI तभी कोई रिस्पॉन्स नहीं देता है, बाकी सब टाइम में ये धड़ाधड़ चलेगा। तो ओके, जो भी है क्या करेंगे अगर वो, अरे बाबू दे इंफॉर्मेशन। डिक, इसका प्रोनाउंसिएशन कैसे?
Dictosome.
गोल्गी मैंने सोच रहा था कि डाइसाइटोसोम होगा, लेकिन ये डाइसाइटोसोम नहीं है। तो गोल्गी apparatus इज ऑल्सो नोन एज गोल्गी कॉम्प्लेक्स और इसे बोला जाता है डिक्टोइजोम। ठीक है, डिक्टोइजोम। It is present in all cell and generally is located close to the nucleus. It appears as flat discs that are dilated peripherally and stacked together in a bunch as a stack of cistern। फिगर 4.8 हमारे पास 4.8 का फिगर है। ठीक है, anatomically and functionally it is closely associated with the endoplasmic reticulum. Functionally, Golgi apparatus two main regions are cis and trans. The region nearer to the nucleus is the cis phase, जिसे cis Golgi बोला जाता है, and the region close to the membrane is the trans phase, that is trans Golgi। तो यहाँ पर इसको स्टैक्स ऑफ सिस्टर्न ने, दीज आर द स्टैक्स ऑफ सिस्टर्न ने। ओके, स्टैक्स ऑफ सिस्टर्न ने, एंड ये होता है आपका पेरिफेरल वेसिकल। स्ट्रक्चर ऑफ अ गोल्गी कॉम्प्लेक्स, नोट द स्ट्रक्चर मेनली consisting of stacks of cisternae। So these are the stacks of cisternae। Okay, this is peripheral vesicle। इसके अलावा और कुछ दिमाग लगाने की जरूरत नहीं। Now function of a Golgi apparatus। Formation of a secretory vesicle। गोल्गी apparatus का काम क्या है? फॉर्मेशन ऑफ अ सिक्रेटरी वेसिकल करना। गोल्गी apparatus इज द साइट फॉर द पैकेजिंग ऑफ अ सिक्रेटरी प्रोडक्ट इनटू द सिक्रेटरी ग्रैन्यूल्स। Material produced in the rough endoplasmic reticulum travels through the lumen of a smooth endoplasmic reticulum। Vesicle boarding of from smooth endoplasmic reticulum transport material to the cis phase of a Golgi complex। Hence, they are also called transport vesicles. Transport vesicles fuse with the Golgi apparatus and empty their content into the Golgi apparatus. Some proteins are phosphorylated and glycosylated here. Also, in the Golgi apparatus, the secretion from endoplasmic reticulum are compacted into highly concentrated packets. These packets then pass to the trans phase where they are packaged into the secretory vesicles. Finally, those small and large vesicles continually break away from the Golgi apparatus and diffuse into the cytosol or discharged outside as cell secretion by exocytosis. The process of cell secretion in glandular tissue happen in 1 से लेकर के 2 आवर. Okay. Now important note. Okay. थोड़ा प्रॉब्लम हो रहा. 1 minute. Important notes. Process of cell secretion in glandular tissue happens within 2 hours. Process of cell secretion in glandular tissue में जो सेल सिक्रेशन का प्रोसेस होता है हैपन विद इन 2 आवर. ओके. When glandular cell is bathed in amino acid tagged with radioactive material, the radioactive protein molecule are detected in endoplasmic reticulum in 5 minutes in Golgi apparatus within half an hour and are secreted to the cell surface in 1 से लेकर के 2 आवर. देन ये तो हो गया गोल्गी apparatus का फॉर्मेशन ऑफ सिक्रेटरी वेसिकल्स. सिक्रेटरी वेसिकल फॉर्मेशन में गोल्गी apparatus सहायता करता है. देन सिंथेटिक फंक्शन. It is the site for the incorporation of carbohydrate into a newly synthesized protein to form glycoproteins. Certain carbohydrate molecules such as a large saccharin polymer cannot be formed in endoplasmic reticulum. These carbohydrates bound with a small amount of protein are synthesized in Golgi apparatus as it occurs in the formation of hyaluronic acid and chondroitin sulfate. Now important notes. Hyaluronic acid and chondroitin sulfate have many functions. Hyaluronic acid and chondroitin sulfate are major component of organic matrix of cartilage and bone. They are major constituent of proteoglycans in the mucus and glandular secretion. They form the ground substance in the extracellular matrix of the interstitial tissue space. They facilitate cell function such as migration and proliferation. Formation of primary lysosomes. Primary lysosomes containing lysosomal enzyme are formed in Golgi apparatus. These lysosomal vesicles extruded from Golgi apparatus are called as Golgi hydrolase vesicles. Figure 4.9. 9 यहाँ पर नहीं है, ओके। यहाँ पर ओके, फ्लूइड मॉलिक्यूल्स, foreign matter being phagocytes, formation of a primary lysosome, secondary lysosome, and a phagolysosome. Formation of a primary lysosome. ओके, बाद में इसको देखेंगे। Transport of a material by discharging material through vesicles, Golgi apparatus helps in transport of a material to other organelles or to other cell surface then replenishes cell membrane. Secretory vesicles formed in the Golgi apparatus are discharged to the cell surface by exocytosis. In the process, the membrane of a Golgi apparatus is incorporated into the membrane of the cell. Figure 4.7 depicts the fusion of the membrane of vesicles with the cell membrane during discharge of secretion by exocytosis. This fusion helps in expanding the cell membrane and replenishing the membrane that is lost or used up for formation of phagosomes during phagocytosis. Thus, cell membrane is continually renewed and replenished by Golgi apparatus. Lysosomes. Lysosomes are membrane-bound spherical organelles that contain a variety of hydrolytic enzyme meant for intracytoplasmic digestion. More than 40 different lysosomal enzyme मतलब lysozyme have been isolated. Some important lysozyme are listed in table 4.2. Lysosomes are found in almost all animal cell except erythrocyte. They are particularly abundant in cell having high phagocytic activity such as neutrophils and monocytes. Important notes. Absence of lysosome. Lysosomes are present in all cell except RBC. They are prominently present in neutrophils. In granulocyte, the lysosome appear as a cytoplasmic granule. Lysosomal activity of a cell or tissue can be determined during the process of phagocytosis. The phagosome that is formed by the cytoplasmic pseudopodia containing foreign bodies such as bacteria, virus, etc. fuse with lysosomes to form phagolysosome, see figure 4.9, and lysosomal enzyme later digest these foreign organisms. Therefore, lysosomes are known as autophagosome. ठीक है, नाउ यहाँ पर एप्लीकेशन बॉक्स 4.3 मार्कर्स फॉर लाइसोजोमल एक्टिविटी. ओके, लाइसोजोमल एक्टिविटी को मार्कर करिए. कौन सा मार्कर यूज है? एसिड फॉस्फेटेज इज यूज्ड एज ए मार्कर फॉर लाइसोजोमल एक्टिविटी. ओके, प्रीवियसली हमने पढ़ा था बायोकेमिस्ट्री में. नाउ important lysosomal enzyme. Table 4.2 important lysosomal enzyme. A प्रोटियोलाइटिक एंजाइम जिसमें कैथेप्सिन आता है जो कि प्रोटीनेजेस होता है. कोलाजिनेज, इलास्टेज. देन लिपोलाइटिक एंजाइम, इसमें लाइपेजेस, फॉस्फोलाइपेजेस, फैटी एसाइल, स्टरेजेस. देन सी में आएगा कार्बोहाइड्रेट स्प्लिटिंग एंजाइम, जिसमें अल्फा ग्लूकोसिडेज, बीटा गैलोक्सिडेज, हायालुरोनिडेज, अराइल सल्फाटेज। डी में आएगा न्यूक्लिक एसिड हाइड्रोलाइजिंग एंजाइम, जिसमें राइबोन्यूक्लियेज आएगा, डीऑक्सी राइबोन्यूक्लियेज आएगा। अदर एंजाइम में एसिड फॉस्फेटेजेस, कैटालेजेस। ठीक है, टोटली ई हो गया। देन द इंटीरियर ऑफ लाइसोजोम इज एसिडिक, मतलब पीएच इज अबाउट 5, 0 से लेकर के 7 एसिड, 7 से 14 बेस। ओके, ठीक है। कंपेयर्ड टू इट्स साइटोजोलिक एक्सटेरियर, मतलब पीएच ऑफ साइटोप्लाज्म इज अबाउट कितना 7.2। एसिडिक इंटीरियर ऑफ दिस ऑर्गेनेल इज ड्यू टू द एक्शन ऑफ प्रोटॉन पंप, द PMH plus ATPase present on the lysosomal membrane that pumps proton H plus from cytosol into the lysosomal interior against its electrochemical gradient. The lysosomal enzyme are acid hydrolases as they function best in acidic pH. Now, type of lysosomes. There are three form of lysosome: प्राइमरी लाइसोजोम, सेकेंडरी लाइसोजोम या इसे एंडोलाइसोजोम बोला जाता है, देन टर्शियरी लाइसोजोम जिसे फागोलाइसोजोम कहा जाता है। फ्लोचार्ट 4.1। प्राइमरी लाइसोजोम, दे आर formed with hydrolytic enzyme synthesized by rough endoplasmic reticulum packaged in the Golgi apparatus. They are generally referred to as a storage vacuoles. Secondary lysosome, they are lysosomes fused with endosomes, hence they are called endolysosomes. Then tertiary lysosome, they are autophagic vacuoles or autophagosomes. They are formed by fusion of phagocytic उसके बाद क्या phagocytic vacuoles with primary lysosome, hence they are called phagolysosomes. Now उसके बाद फ्लोचार्ट 4.1 टाइप्स ऑफ लाइसोजोम एंड देयर फंक्शन। लाइसोजोम तीन प्रकार का है: प्राइमरी, सेकेंडरी, देन टर्शियरी। प्राइमरी लाइसोजोम गोल्गी हाइड्रोलेज वेसिकल, सेकेंडरी लाइसोजोम एंडोलाइसोजोम, टर्शियरी लाइसोजोम फागोलाइसोजोम। देन ये प्राइमरी लाइसोजोम न्यूली फॉर्म्ड लाइसोजोम होता है, एंजाइम इनएक्टिव होता है। सेकेंडरी लाइसोजोम प्राइमरी लाइसोजोम फ्यूज्ड विथ एंडोजोम, एंजाइम आपका एक्टिव रहता है। टर्शियरी लाइसोजोम, लाइसोजोम कंटेनिंग डिग्रेडेड प्रोडक्ट आफ्टर फागोसाइटोसिस, नो एंजाइमेटिक एक्टिविटी सीन। चलता है important notes. Residual bodies, after the material in phagosome have been digested by lysosomes, some waste material may be left. The undigestible remnant in the lysosomes are called as residual bodies. Residual bodies containing indigestible substance are excreted from the cell by exocytosis. Function of lysosomes. Digestive organ of the cell. Function of lysosome. Digestive organ of the cell. Lysosome contain many enzymes essential for intracellular digestion. Absence of these enzymes lead to various storage disease. Clinical works 4.2 as listed in table 4.3. Bactericidal activity. Lysosome kills and remove ineffective organisms and foreign bodies. Lysosomes are highly bactericidal. Mechanism of killing of bacteria by lysosome include lysozymes that dissolve cell membrane of bacteria. Lysoferrin present in lysosomes binds iron and other substance before they can promote bacterial growth. Then acidic pH of about 5.0 in lysosome activates lysosomal hydrolases and inactivate bacterial metabolic system. Autolysis. When a cell is damaged or ruptured, the enzymes liberated within the cytoplasm digest the cytoplasmic content, a process known as autolysis. The autolytic enzymes are mainly lysosomal enzyme. Important notes. Autolysis has two important functions. Regression of a tissue, few tissue in the body regress to a smaller size. For example, regression of a uterus after pregnancy, atrophy of a muscles during long period of inactivity, and decreases in size of a mammary gland after the period of lactation. Lysosomal enzyme facilitate this progression process of regression through the mechanism of increased lysosomal activity in this tissue is not clearly known. Second one, removal of a damaged cell or tissue. During damage to the cell caused by mechanical, thermal, chemical, or any other injury, lysosomes are ruptured and hydrolyses are released that digest the cell material and surrounding organic matrix. The damaged cells are digested by the process called as autolysis and are completely removed. Later, the new cells formed by mitotic reproduction from adjacent cell replace the old or damaged cell. Now table 4.3 common lysosomal storage disease. नंबर वन डिजीज कॉलम में फाब्री डिजीज. फाब्री डिजीज एंजाइम डेफिशिएंसी अल्फा गालाक्टोसिडेज ए. यूनिक फीचर होची क्यूटानियस एंजियो केराटोमास एंड हाइपोहाइड्रोजेस. देन गाउचर डिजीज इसमें एंजाइम डेफिशिएंसी होता है एसिड बीटा ग्लूकोसिडेज. यूनिक फीचर होता है हेपाटोस्प्लिनोमेगाली एंड स्केलेटल डिस्प्लाजिया. यूनिक फीचर. देन नीमैन पीक डिजीज इसमें स्पिंजोमाइलिनेज एंजाइम का डेफिशिएंसी होता है. यूनिक फीचर होता है मेंटल रिटार्डेशन सीजर लॉन्ग फेल्योर. देन टेसाक डिजीज जिसमें बीटा हेक्सो सामिनीडेज ए. इसमें भी मेंटल रिटार्डेशन होता है मैक्रोसेफाली एंड हाइपर एक्यूसिस इन इन्फेंट्स. देन हुर्लर डिजीज इसमें अल्फा एल मतलब लिभो इडुरोनिडेज इसमें मेंटल रिटार्डेशन. कोरेज फेसिस सीवीएस कार्डियोवास्कुलर सिस्टम इन्वॉल्वमेंट होता है. देन पॉम्पी डिजीज इसमें एसिड अल्फा ग्लूकोसिडेज होता है एंड इसका यूनिक फीचर में आपका आ जाएगा मायोकार्डियोपैथी भी होता है. देन अगर हम देखेंगे फंक्शन ऑफ लाइसोसोम की हम बात कर रहे थे तो उसका 4 नंबर पॉइंट हो जाएगा रिसाइकलिंग ऑफ सेल ऑर्गेनेल. द ओर्न आउट सेल ऑर्गेनेल आर ट्रांसपोर्टेड टू द लाइसोसोम बाय ऑटोफागोसोम्स and are then transferred into the interior of lysosomes. Lysosomal enzyme digest these organelles and the process is known as autophagy. However, in this process, the nutrients liberated from digested organelles are reused by the cell. This is one among the several mechanisms of cell survival during scarcity of nutrients. इम्पोर्टेंट नोट्स क्या है. ऑटोफैजी इज ए हाउस कीपिंग फेनोमेनन ऑफ द सेल. ऑटोफैजी मतलब टू ईट वन सेल्फ मतलब खुद ही खुद को खा जाना. सो ऑटोफैजी बाय लाइसोसोम्स इज द प्रोसेस ऑफ किलिंग and degrading the degenerating and obsolete organelles of the cell. Also, large protein aggregates are degraded and recycled. Thus, autophagy is a housekeeping process of the cell. Autophagy contribute to routine turnover of cytoplasmic components. Then fertilization of ovum. Acrosomes located on the head of a spermatozoa is a specialized lysosome that contains many hydrolytic enzymes. Acrosomes play an important role in the penetration of ovum by sperm. Then removal of a product of a metabolism. Lysosome removes intracellular product of a metabolism from the cell. Therefore, in the lysosomal storage disease, deficiency of a specific enzyme leads to accumulation of these metabolic products in lysosomes and inclusion bodies in the cell that causes cell dysfunction. Then clinical works 4.2, lysosomal storage disease. Lysosomal storage disease, clinical box 4.2. Congenital absence of a lysosomal enzyme leads to accumulation of a material in the lysosome that are usually degraded by the lysosome, causing lysosomal storage disease, especially elimination of a certain substances from the cell of the body. Example, glycogen, cerebrosides, ganglioside, and sphingomyelin mainly depends on the activity of a lysosomal enzyme. Therefore, in the absence of active lysosomes, intracellular accumulation of these substances interfere with normal cell function and produce disease. Accumulation of a partly digested cellular material is called inclusion bodies. Then ribosomes. Though ribosomes are usually present on the surface of endoplasmic reticulum, they are also present as free organelles in the cytoplasm. They contain about 85% of RNA of the cell. They are the major site of protein synthesis. RNA is also present in the nucleus, mitochondria, and cytoplasm, and consequently protein synthesis occur in this structure too. They may be present singly मतलब monoribosome or in groups मतलब polyribosomes. Then coming to the peroxisome. ଆଉ କେତେଟା ପେଜ ଅଛି? ଦୁଇଟା. Then coming to the peroxisomes. Peroxisomes are small spherical organelle having diameter of about 0.5 micrometer and therefore denoted as microbodies. Specialities of a peroxisome. They are formed by budding from or by division of a smooth endoplasmic reticulum. They are referred to as subcellular respiratory organelle, but they do not have energy coupled electron transport system. They contain oxidases that promote oxidation of lipid, especially omega oxidation of long chain fatty acid, forming acetyl coenzyme A and hydrogen peroxide H2O2, and catalases that liberate oxygen from hydrogen peroxide. They protect the tissue from oxidative stress. Application box 4.4. Peroxisome protect from oxidative stress मतलब OS. So hydrogen peroxide, a potent oxidant produced during metabolic reaction, is one of the agent for oxidative stress. Hydrogen peroxide is destroyed by catalysis, a peroxisomal enzyme, which is designated as an antioxidant for combating oxidative stress. Thus, peroxisome protect cell from toxic effect of a hydrogen peroxide. Hence, increased catalase activity is one of the marker of oxidative stress. Number 3. With the help of peroxine, the protein capirons various protein with specific signal sequence are directed to peroxisomes. The membrane of a peroxisome contain a number of specific protein that transport substance between peroxisome matrix and cytosols. Peroxisome matrix contain more than 50 enzymes that are involved in many metabolic reactions of lipid, long chain fatty acid, plasmalogen, cholesterol, bile acid, purines, amino acid, hydrogen peroxide. Deficiency of enzyme leads to many disease. Clinical box 4.3 में देखोगे. Proliferation of peroxisome is caused by many synthetic products that act on a receptor in the nuclei of the cell known as peroxisome proliferation activated receptor, that is PPARs. PPARs are a member of a nuclear receptor superfamily. Those on activation bind to the DNA and produce change in messenger RNA production. PPARs have extensive physiologic effect and affect many tissue and organ. Clinical box 4.3 में क्या है? डिजीज ऑफ पेरोक्सिज्म. पेरोक्सिज्म में बी abnormal or absent in inherited disorder, that is called to Zellweger syndrome, occurs due to the mutation in the gene encoding certain protein such as peroxin and peroxisomal enzyme. This syndrome is characterized by profound neurologic impairment, accumulation of a very long chain fatty acid, abnormalities in synthesis of a bile acid, and marked reduction of a plasmalogen. The child usually dies within a year in this disorder. Another similar genetic disorder is infantile refsum disease, which is less severe and only few proteins are affected. Then Brown slider disease, adrenoleukodystrophy, occurs due to the insufficient oxidation of a very long chain fatty acid by peroxisomes. This is an autosomal recessive disease that manifests with progressive degeneration of liver, kidney, and brain. Primary hyperoxaluria occurs due to the defective peroxisomal metabolism of a glycosylate derived from glycine. सो आज के लिए इतना ही, फिर मिलते हैं नेक्स्ट. यहाँ से हम क्या करेंगे, इसको एंड करेंगे. यहाँ तक हमने आज पढ़ा है, ठीक है. यहाँ पर हम इसे एंड करेंगे. डेट भी मेंशन कर देते हैं. आज शायद 9 डेट है, ठीक है. जब भी स्टार्ट करेंगे 9/1/26. Thanks for watching.